Hello to everybody. It's a pleasure to participate in this conference, and I would like to thank the organizers of the meeting for inviting me to present our results at this conference, and my special thanks to Professor David Caresi. My presentation is entitled as Quantitative Genetics and Omics Study of Chronic Widespread Pain Syndrome. As one can see, I am only presenting this study on behalf of many people who collaborated and contributed very substantial into development and progression of this project, including colleagues from my department as well as colleagues from King's College. The study results were summarized in a number of the research papers which were published during the last 5 and 6 years, and some of them are presented on this slide. So our research is contributed to genetic analysis of chronic widespread pain, a medical disorder which can be found actually above and below the waist and on both sides of the body, with prevalence which can reach 10 to 15% in the general human population in developed countries. It has been considered as a musculoskeletal disease, fibromyalgia, but also often considered as a neuropsychiatric condition. CWP characterized by continuous chronic pain and painful response to pressure, and they are accepted by American College of Rheumatology criteria, which include several parameters, for example, durations of more than 3 months during the year, or 11 of 8 and tender points under the pressure of 4 kilograms, and several others. This is clearly multifactorial, complex multifactorial condition, and the published data suggests that CWP have undoubtedly complex etiology, including contribution, of course, age, sex is much more prevalent among females, lifestyle, inflammatory, metabolic, genetic, and epigenetic factor. And this slide on the right side clearly shows its increase with age. As I say, this is in particular common among females. In this context, or in context of our research interest, the major question which was of interest for us, whether and to what extent this condition depends on the genetic factors and where these genetic factors could be located in our genome. The study included a number of populations, of which the major one was the discovery sample coming from the UK, from the King's College, UK, so-called UK twins. More than 2,500 individuals are included in this research project, assessed for many variables that will be mentioned in the future. The replication sample was of approximately the same size, but independently unrelated individuals from Germany, so-called core research project. And recently, the results of the study that we obtained, we validate, checked, and replicate using the big data, in particular UK Biobank, one of the probably largest biomedical databases in the world, containing more than 500,000 individuals who were genome-wide, genotype, and for whom a lot of health information is available. The approach and design of the study is rather complex, and presenting this in a very schematic way would look like the first stage, including the data collection, which is, of course, recruitment of participants visiting to King's College for complex assessment, including, of course, detailed assessment of the pain protocol and blood samples collection, the assessment of body compositions, and many other features. At the next stage, when conducted the whole genome genotyping using at that period of time when we started the study approximately 2.5 million SNPs, including, of course, imputed markers. Similarly, we conducted the whole epigenome study, implementing the DNA methylation analysis of roughly 11 million methylation size clone beings. The metabolomic assessment was also the part of the research project, together with other measurements, and the metabolomics assessment that was conducted approximately 6 years ago, included 510 metabolites. All this huge amount of data was conducted extensive statistical and bioinformatics analysis. The first and the major question which was interested as and for me as in genetics, it was of particular interest, is what is the familial or genetic component, whether there is a familial or genetic component in the CWP. And the answer is yes, there is. The familial aggregation of the CWP, as estimated by tetracorrelation coefficient, was statistically significant and well compatible with the Mendelian expectations, as we see substantially higher correlation between the monozygotic twins in comparison to dizygotic twins. That, of course, we were interested also in more accurate assessment of the contribution of the putative genetic factor heritability. And because this is the binary phenotype, we were interested in contribution of genetic factors into liability to CWP, first of all in our discovery sample. And since our sample consisted of monozygotic and dizygotic twins, we could implement family-based variance component analysis. However, this is binary phenotype, and therefore the classical variance component analysis is not that much appropriate for this. The CWP, as many other phenotypes, is dichotomous, binary, yes versus no, and therefore it can be treated if this binary phenotype has underlying quantitative basis liability, expressed as a threshold phenotype. In our group, Dr. Ida Malkin, first of all, developed the method based on Douglas Falconer's polygenic threshold concept for dichotomous phenotypes and inheritance. And we developed a new software to examine the extent of genetic influence on CWP liability under the age and other covariates dependence. The model is quite complex. We will not go in details for this, but our results were clear-cut. It showed that despite the simultaneous adjustment for the effect of age, fatness, smoking, the contribution of the putative additive genetic factors was quite substantial and highly significant. This is the standard error. Interesting to mention also that this software, this method allows us to conduct the variance component analysis, including both binary phenotypes. So one is the binary phenotype, and the second one, as in this case, the Friday score quantitative phenotype, and to estimate a possible additive correlation between the liability score for the one phenotype and the variation of the second phenotype, as well as the common environmental component. So at the first stage, our conclusion was that the preliminary data showed that chronic widespread phenotype syndrome is a common condition in general human population. Its prevalence increases with the age. Our quantitative genetic analysis suggests significant involvement of the genetic factors in the manifestation of CWP and associated Fried phenotype. The next question that raised in in this relation was what are the specific molecular genetic factors that cause this familiar aggregation and high estimates of the particular genetic factors affecting the CWP. The design of the next stage, including the analysis of the whole metabolome, genome, and epigenome, as you see on this diagram, related to one another. And of course, the major point is once the results are obtained, whether they could be reproduced in other samples. The results of metabolomics studies show the number of the metabolites that were highly significantly associated with the presence of CWP versus non-affected individuals. But in particular, interesting results were obtained with the molecules related to androsterone metabolism and show that most significant association between the epi and the hydroepiandrosterone sulfide. Okay, these two. Here are the p-values. So these estimates were extremely high. These results were reproduced quite accurately in core sample. And when we tested, for example, using the GWAS, the association of the CW, when we tested the association, conducted the GWAS between the between the variation of the metabolites of interest, in particular epiandrosterone, we in both samples find a highly significant association between the specific genomic region and the epiandrosterone variation, which was extremely interesting. Next, when we conducted Mendelian randomization analysis, we got some evidence. They are not yet clear cut. The sample size is not substantial enough, but nevertheless, there are indications that epi or dehydroepiandrosterone sulfate could be among the causative factor for CWP. We next conducted the GWAS of CWP itself, of course, the functional genomic annotations and gene ontology analysis to estimate the possible enrichment, enrichment of some particular aspects of the genome. And the results were quite interesting. For Go analysis, we used approximately 1,400 genes that have been simultaneously and nominally significantly associated with our phenotype in both our samples, and the combined probability for in both samples was below 0.001. We set up a threshold for GOP values as P001 and examine two domains or Go domains, biological processes and cellular component. And we find that the 10 most significant Go terms for BC and CC were related to neurological system and sensory function, which is very interesting. And the specific definitions for these terms were like neuron recognition, synapse assembly, presynaptic membrane organization, synapse organization, neural part, postsynaptic density, neuron projection, and so on and so on and so on. And several genomic regions were of particular interest and under our consideration currently. This, for example, includes the synapsin gene encoding neuron phosphoproteins, which associated with cytoplasmic surface of synaptic vesicles. And our conclusion at this stage was implementing metabolomic analysis in combination with GWAS. We observed highly significant association of several androsterone-related endogenous hormones with risk to CVP. Further gene ontology enrichment analysis suggested that genes related to neurological system and sensory function clearly overrepresented in GWAS in both our samples discovery and in replication samples. We therefore proceeded to the next stage, which included the epigenomic study. The design of epigenomic study was quite complex, as can be seen from this picture. And again, because of the lack of time, we cannot go into too many details, but may say that at this preliminary stage, our sample was divided into relatively independent samples. One was including 50% discordant MZ-genes, and the second one included 1,600 unrelated individuals. And what we observed, the measured results could be formulated like this. We first of all did observe that approximately 700,000 methylation sites were longitudinally stable, that is to say, correlated significantly in the repeated measurement analysis. When the measurements were taken one from another in several months' time. Of these 700 beans, approximately 41,000 exhibited non-significant association with CVP, with signals at the genome-wide suggestive level. And again, we conducted the gene ontology analysis and compared the results obtained in GWAS and EWAS, mostly in our discovery sample, because this information was not available from other samples, including coral. The comparison of the results obtained in EWAS were very similar to what was observed in GWAS, and we again received the indication on the involvement of the same genomic regions that are related to neurological system and sensor function. The most significant functional genomic regions were defined as neuron recognition. In biological process category, we mapped several genes which are indicated here, and cellular component again, we find that number of genes fell in the category of neuron projections, and the major genes were like this, and they were common for both GWAS and EWAS, which is very interesting. We got the further confirmation to this by continuing to analyze using the bioinformatics method, and so we come into conclusion that genomic regions associated with nervous system dominate a list of the potential candidate genes that will come out from this comparative analysis. So we can conclude now that implementing the whole epigenome methylation analysis consistently revealed approximately 41,000 methylation sites nominally significantly associated with CWT, and further ontology analysis suggested the genomic regions related to neurological system and sensory function similarly to GWAS results in overrepresentation of these genes in the array of methylation beings identified. And the last point that I would like to mention quickly now is the question whether the obtained results could be reproduced or confirmed in the big data, in particular in UK Biobank. And what you see here is one of such analyses that include polygenic risk scores. And the upper part is conducted within this one population, but the second part is the two sample study in which UK Biobank data were used as base for replication in twins UK sample, and we tested two phenotypes, low back pain and CWP, again in UK Biobank, and some results appeared as an interesting results and reached the statistically significant level. So I believe at this stage I can make my final conclusions and to summarize everything that was presented above. First, the results obtained in the present study show that CWP has a substantial familial component, which is at least partly could be attributable to genetic factors. The latter include at least two categories of biologically active molecules. The first, those related to androsterone metabolism as suggested by our metabolomics analysis, and those related to neurological system and sensory function as suggested by GWAS and EVAS and corresponding bioinformatics analysis. The major results observed in our discovery sample were reproduced and corroborated in our replication sample as well as partly in big data. So this is the ongoing project, and I would like to thank you all for your attention and time. Thank you.